Hey there, I'm a recent university graduate who's attempting a new venture to pique interest in the fascinating world of nucleic acid research, which I fell in love with during my graduate school. I have a lot of content that is planned for this channel, but I'm starting with a short 3-part introductory series to acquaint people with this field. In this first video, we will look into what are nucleic acids, what is the central dogma of molecular biology, what are nucleic acids made of, and the structure of a DNA. When we zoom down from the magnitude of the Earth, which is in the thousands of kilometers, to human beings, organ systems, and down to the level of a DNA, it is around a micrometer in length when condensed. Fun fact, when the DNA is unraveled, it can be up to 2 meters long. This is the amount of information that a single DNA can hold. The condensed DNA that is tightly packed inside the cell is known as a chromosome. Sections of these chromosomes that encode information for the cells to function are known as genes. The DNA resembles a hard disk that contains softwares that perform various tasks that are essential for a living system. RNA is like a single software that is read from the master DNA. This process is codified in the central dogma of molecular biology that states that the DNA is read into RNA through a process known as transcription, and then the RNA is translated into protein. When a cell divides, the DNA is replicated so that each of the daughter cells can have its own copy of the DNA. In some viruses, RNA can be reverse transcribed into a DNA by a process known as reverse transcription. This DNA is known as cDNA or complementary DNA. It is also the underlying principle of an RT-PCR test, which was prevalent to detect active infections during the peak of the pandemic. The biomolecules DNA, RNA, and proteins are known as biopolymers because they are made of repeating units called monomers. It is also safer to call the product of translation as a polypeptide that is made of monomers called amino acids. A polypeptide is considered a protein after it has folded into its proper form by further modifications. The monomer units that make a DNA and an RNA are known as nucleotides. These are made of a 5-membered sugar called pentose sugar, nitrogenous base, and the phosphate group. The pentose sugar in a DNA is deoxyribonucleotide, and that in an RNA is ribonucleotide. Hence the names deoxyribonucleic acid and ribonucleic acid. There is a distinction between the DNA and RNA monomers, as you may have noticed. Yes, the hydroxyl group in the 2' position of a DNA is missing. As a result, a DNA is usually more stable than an RNA. We will cover this in more detail in a future video. There are 4 distinct nitrogenous bases for each of the polymers DNA and RNA. In DNA, these are adenine, guanine, cytosine, and thymine, with uracil serving as a substitute for thymine in RNA. Adenine and guanine are known as purines, whereas cytosine, thymine, and uracil are known as pyrimidines. Let us take the example of guanine. The 3 highlighted groups are required for the formation of hydrogen bonds, which give DNA its distinct double-stranded form. Canonically, purines always pair with pyrimidines, so adenine and thymine form 2 hydrogen bonds, while guanine and cytosine form 3 hydrogen bonds. As a result, adenine and thymine, as well as guanine and cytosine, are complementary bases respectively. Adenine also pairs with uracil in an RNA, but RNA is usually single-stranded, therefore it pairs internally. We will cover more about RNA structures in the next video. To bind with its complementary base on the sister strand, canonical base pairing is used. The monomers are joined by covalent bonds that are formed between the 5' phosphate group of the next nucleotide in the series and the 3' hydroxyl of the previous nucleotide. As the monomers are built from the 5' end towards the 3' end, it looks like the DNA double helix that you see on the right side. Over here you see the double helical structure of the DNA that was proposed by Watson and Crick, which was based on the X-ray crystallographic observation made by Rosalind Franklin and Raymond Gosling that you see on the left side. But the DNA structure that was visualized by Rosalind Franklin was an A form of the DNA, which differs from the more ubiquitous B DNA. It is understood that A DNA is more readily observed under experimental conditions in the lab when the DNA is dehydrated. However, researchers have also found A DNA in many natural systems. Each form of the A, B, and Z forms of DNA can exist naturally inside the body when influenced by external factors. In summary, in today's video, we saw that the DNA is the primary information carrier of all living systems. The mRNA message is read from the DNA and then translated into a protein. The different monomers that make up a DNA, and we saw what were the differences between a DNA and an RNA, especially in terms of the nitrogenous base and the 2' hydroxyl group in the sugar of the ribonucleotide. We also understood the basic idea of how a DNA polymer is constructed from its monomer, the discovery of the DNA double helix model, and we saw differences between the different forms of DNA. To end this video, please find a small concept check here for revision, and thank you again for watching the video and hope to see you in my next video. Till then, please like, share with your friends and subscribe to be notified of new content. Thank you very much. Bye.